Good morning and welcome to our Friday morning session for Cosyne 2021. I'm Anne-Marie Oswald and I'll be the moderator for this session, and we have some very exciting speakers to meet this morning, and I'm going to just start off and get us rolling right away here. Our first speaker is Anne Duan. She is from the Chinese Academy of Sciences in NYU Shanghai. Her talk this morning is entitled Neural and Computational Mechanisms of Task Switching. And I will leave that to you, Anne. Take it away.
Thanks, Anne-Marie, for the introduction. Let me try to share my screen. Does it look okay? Okay. Hello, everyone. Thanks to Cosyne for selecting our work for a talk. I'm currently a postdoc in Xun Yilong's lab at the Institute of Neuroscience in Shanghai, where I actually study cortical-collicular contribution to motor planning in high-fixed mice, very different from what I'm going to talk about today. If you're interested in that line of work, you can check out the remnants of Yuxing's poster from yesterday. But today, I'll talk about task switch cost. This is a collaboration that I have with Jeff Erlich, built on data that I collected in the Brody lab. After Jeff started his own group in NYU Shanghai, he and his students, Xiangci Li and Ma Ce, continued the investigation using recurrent neural networks, with input from Zheng Zhang, who is now the director of Amazon Shanghai AI Lab. Okay, so what is task switch cost, and why should we care to study it? In our daily life, we often have to rapidly switch between two familiar tasks. I switch between speaking Chinese and English multiple times a day, and sometimes it can be surprisingly difficult, even though I am fluent in both languages. I can be slower and make more mistakes right after I switch. This kind of phenomenon is actually quite prevalent, especially when there's a high degree of conflict between the tasks. And switch costs can be studied in the laboratory using behaviors such as the Stroop task, where subjects are instructed to either read the text of each word or name the color of each word. For example, this word corresponds to orange in the word reading task, but to blue in the color naming task. Subjects can switch between these tasks on a fast sub-second timescale. So this is different from reversal learning. But there's a cost when they switch, with increased error rates and increased reaction time immediately after the switch. So how does switch cost emerge, and what is the underlying mechanism? To answer these two questions, we use two complementary model systems, rats and artificial recurrent neural networks. I'll first start with our earlier rat work that motivated this study, and then move on to what we have learned from parallel investigation using both systems. To study task switching, we trained rats on the pro-anti task switching behavior. On each trial, rats are presented with an auditory cue that indicates the task rule for that trial, pro or anti. Rats then have to remember the task rule over a silent delay period, after which our target stimulus will be presented as either the left or the right light. If it's a pro trial, rats should orient towards the light for reward. If it's an anti-trial, rats should orient away from the light for reward. And there's an inter-trial interval between trials. In each session, pro and anti-trials are presented in alternating blocks, but there is the task rule is explicitly cued on each trial. So is there a cost when rats switch from one task to another, as observed in analogous primate behaviors? Indeed, we found a robust task switch cost in rats. Shown here is rats' performance right before and after task switching. And you can see that there's a significant drop in accuracy on the first trial of new blocks. We also observed increasing reaction time on switch trials. So the difference between switch and block trial performance of the same task rule is what we call switch cost. But please note that the accuracy on switch trials is still significantly better than if the animals were only relying on reward feedback for task switching, as in reversal learning behavior. So the animals are monitoring the task queue on each trial, but there's a cost when they switch. How do we explain such a cost? One major theory posits that leaving the old task is difficult. Forgetting the old task is difficult. So the residual memory of the old task is the source of interference that results in a switch cost. Previous behavior studies have reported inconsistent results regarding the mechanism of task switch cost, and direct physiological tests of these theories are yet to be done. So here we set out to test 3 predictions of the residual interference theory of task switch cost. First, the theory predicts that there should be a representation of the old task before task switching. Second, stronger residual memory should lead to stronger interference and therefore larger switch cost. And finally, causal manipulation of this residual memory should change the subsequent switch cost. To test these predictions, we want to use rats and RNNs in complementary ways. But before we get there, first we ask, do artificial neural networks develop switch costs when trained to task switch? We train RNNs in the parental behavior using the same trial structure and shaping steps as we use for rats. RNNs are element recurrent networks consisting of an input layer, a recurrently connected hidden layer, and an output layer. Each trial of RNNs is divided into 5 time steps corresponding to the 5 behavior epochs for rats. And the weights are updated using backpropagation through time. Similar to rats, we started RNNs on pro-only training. And after they get good pro performance, we then move on to within session and pro-anti-task switching. So RNNs displayed similar learning curves as rats, but if we keep training them, eventually they'll be perfect in their performance. So we actually stopped the training when RNNs achieved comparable performance to rats, and then we analyzed RNNs' behavior and dynamics with frozen weights. I should emphasize that RNNs are only optimized for good pro and anti-performance, and they're not trained to have a switch cost. So we were actually surprised to find that, just like rats, RNNs develop task switch costs. You might be wondering, it looks like the rats have a switch cost asymmetry, but the RNNs do not. I can come back to that in the question period. In RNNs, further training reduced task switch cost because eventually they are perfect on every trial. But this inspired us to look back at the rat data and found a similar trend where switch costs reduce with further training, even though to a lesser extent. So we're wondering what aspect of the training did the trick? Is it because agents became better at each task, or they gained more experience on task switching? To test that, we controlled the total number of training trials. We held that constant, and then varied the block length during training, and found that shorter blocks, aka more practice on task switching, resulted in a smaller cost. So practice on switching is really the key here. So far, we found that switch costs can emerge in biological as well as artificial agents, so it does not require specific neural constraints. Instead, switch cost likely results from a lack of experience on task switching. Importantly, these data suggest that we can use RNN as a complementary model system to study mechanism of task switch cost. So now we can test the predictions listed before. We analyzed population activity from prefrontal and midbrain regions of trained rats and from hidden units of trained RNNs. In rats, single neurons encode pro-anti-task information, as shown here, by different firing rates on pro versus anti trial across epochs. And as usual, neurons display heterogeneous patterns, and these signals are distributed across the brain. In a separate manuscript, we actually study how features of single neurons together contribute to sensory-motor routing in a pro and anti-task, but not on switch trials. We have very few switch trials per session, so to study them, we have to rely on single trial population level analysis, which is the approach that we're taking here. We included simultaneous recorded single and multi-units, and then denoise the population activity using principal component analysis. We included top PCs that explain 95% variance on correct block trials. We then trained classifiers on the denoise population activity to differentiate activity on pro versus anti trials and define a task rule dimension. Finally, we project the activity on left-out single trials onto the rule dimension to obtain an estimate of the encoded task rule on these left-out trials. We call it the rule score, which indicates the direction and strength of the rule encoding on these left-out trials. So this method allowed us to effectively read out the population level task rule encoding on single trials from rat and RNN dynamics. So are these signals purely sensory signals, or do they actually influence behavior? We examined neurobehavioral correlation on a trial-by-trial basis, first focusing on block trials. So the dots and lines are the data and fit to percent correct as a function of same rule score, transformed from rule score so that the positive values mean encoding of the current task, and negative values mean encoding of the opposite task. And you can see that rule scores strongly predict rat's choice accuracy. And we fit a Jiaolan model to quantify this correlation per epoch per session, and then plot the distribution of coefficients here. So in both rats and RNNs, we found that population level task rule encoding is significantly correlated with behavior in all task epochs, even during the intertrial interval. So think about what this means, right? This means that within a rule block, the rule information in the ITI facilitates performance on the next trial. Pretty interesting. So I want to implant a seed in your brain right now. So these data with block trials sort of inspired us to hypothesize that maybe the lower accuracy on switch trials, aka switch costs, can be explained by the weaker rule encoding. So plot here are RNN same rule scores on switch versus block trials. So now we're looking at switch trials. And you can see that indeed, switch trials have weaker task rule information compared to block trials in all task epochs. What about the intertrial interval? Is there a representation of the old task in the ITI before switch trials? Again, we're plotting same rule scores, so positive values mean representation of the current task, and negative values mean representation of the old task. So consistent with the residual interference theory, we found a representation of the old task in ITI before switch trials. And rats show very similar patterns, except that biology is noisier than machines. More importantly, we want to know if stronger residual interference from the old task will lead to worse performance on switch trials. So again, linking back to behavior. Plotted here in dotted lines are percent correct on switch trials as a function of ITI rule scores before switch trials. So you can see that the stronger the residual memory of the old task set, the worse the performance on switch trials, which means the larger the switch cost. Again, consistent with the theory. Finally, let's do some in silico causal experiments to see if manipulating this residual memory can change subsequent switch cost. We first tested necessity by silencing IT activity of RNN hidden units before switch trials while preserving the input of the new task queue. Shown here is relative switch cost as a function of IT activity, ranging from normal activity to complete silencing. And you can see that indeed, selective silencing of IT activity reduce task switch costs correspondingly. Conversely, we asked if inserting a memory of the opposite task rule before block trials was sufficient to generate a switch cost even in the absence of task switching. So what we did here is we systematically mixed the IT activity before block trials of opposite tasks. And you can see that complete switching of IT activity resulted in a comparable cost on block trials, and different mixtures resulted in intermediate levels of cost. So together, the population analyses as well as the perturbation experiment suggest that the memory of the opposite task rule carried over from the previous trial interfere with the encoding of the new task, in turn generating a switch cost. You might be wondering, what about in vivo causal test of the theory? I can come back to that in the question period. But before I go, I want to revisit the question of why. Why did rats and RNNs develop a strategy that results in a switch cost? So it seems like, at least our data suggests that on block trials, agents partially rely on block identity to help compute choice rather than encoding the relevant task rule on every single trial. This strategy exploits the stability of the task environment within the block, but sacrifices some flexibility during task switching. Thinking more broadly, in an environment where there are both long-term regularities as well as short-term changes, agents need to take advantage of the stability while maintaining flexibility. Flexibility is great, but it's computationally expensive. So sometimes agents opt for reduced flexibility by relying more on well-practiced actions and habitual behaviors. And similar mechanisms have been used to explain serial biases in the form of sensor history or choice history, as well as how environmental volatility could modulate learning rates. Maybe the final take-home from our study is that if you want to be good at task switching, you should practice task switching, because being too much in the groove for one task could actually put us in a state that reduces flexibility. This might be a good segue for my upcoming lifestyle of perpetual multitasking. I'm happy to share that I'm starting my own group at Sainsbury Wellcome Center, which is an amazing place to do science. So if you're interested in understanding how animals make value-based decisions under risk and uncertainty and how they behave in multiplayer games, please get in touch. We're hiring students and postdocs. Finally, thank all of you for your attention, and I'm happy to take questions.
All right. Very excellent talk. Congratulations on the new position.
Thank you.
We've got a few questions coming in from the audience here. I'm just giving a little bit of chance here for some population here. But there's a good question here from Kara Weisman. She says, it looks like the cost from pro to anti is different in magnitude from anti to pro. And she said, I forget which one's larger, but why is that?
Yeah, that's a really good question. Let me see. I have a slide prepared for that. So you're totally right that there's an asymmetry in the rats, right? So the cost of switching out of the harder anti-task into the easier pro task is larger. So I want to say that in Stroop task for humans, there's a similar trend where switching out of the harder color naming into the easier word reading has a larger cost. So this is, again, consistent with the task inertia theory, which is when you are in a harder, more cognitively demanding task, the inertia is bigger. So leaving that and going to the new task is harder. I do want to comment on how, even though we trained RNNs with similar shaping steps, we didn't actually see the asymmetry here. You can see at least no consistent asymmetry. So we think that part of the reason is because animals have a lifelong experience that may increase the asymmetry between pro and anti-task, right? For example, as humans, we read words way more often than we name colors. But for RNNs, the only experience they have is the experience we give them, which is task training. So we want to ask if we can create a switch cost asymmetry in RNN by creating a more extreme asymmetry experience, and the answer is yes. So as shown here, we are controlling the total number of switches, but we vary the ratio of pro to anti switch. And you can see if you have a lot of experience on pro to anti switches, we replicate the switch cost asymmetry as in rats, right? So this is what we see in rats. But more importantly, we can create the opposite asymmetry by manipulating the experience. So I think the take home here, and maybe to answer your question, is that the experience is really powerful, both in terms of shaping the switch cost, but also the switch cost asymmetry.
Okay. Our next question is from Christine Constantinople. She says, great talk, Anne. Did you train the recurrent neural net on a version of the task in which pro and anti trials were interleaved? Was there a rule in coding during the ITI?
Yeah, that's a good question. I think one way to answer that question is that so when you see the when we vary the block length, right, that's kind of going in that direction. The shorter the block length, the smaller the switch cost. So in animals, we have trained them on the interleaved version of the task, and the pattern of switch cost is very different. So for RNN, we were trying to match the animal's training, so we did not train interleaved version. But I think that based on our current data, I will predict sort of a smaller switch cost.
Okay, and I think that basically just answered Cooper Grossman's question as well. He wanted to ask the relative variance between the short block sessions versus long block sessions, and I think you just answered that. So from Yulong Li, I think you said practicing task switching makes switching easier. Is the task memory during ITI also the mechanism of that?
Is the task memory during the ITI a mechanism for that? So I think there's several ways of thinking about this, right? Like one important thing is that we were surprised to find that for block trials, the ITI memory helps animals' performance, right? Like that was kind of the slide I showed where the ITI root score is correlated with behavior on the next trial. So ITI memory facilitates performance for both rats and RNNs within a block. And for the same mechanism during task switching actually hurts them. That's the sort of the residual interference I'm talking about. In terms of the practice thing, yeah, so that's really interesting because switch trials occur less frequently compared to block trials. So at the beginning, both rats and RNNs have very few experience, not as much experience with switch trials, but as they practice the switch trial, they have more experience. So it would be interesting to sort of, I think what you're asking is that, can we then dissect the RNN dynamics to see if as training goes on, the memory goes away or in a way that like doesn't affect them. We haven't really directly looked at that, but that would be an interesting thing to do.
Okay, well, we're going to have to move on here. Thank you, Anne, for a very nice talk. Let's bring in our next speaker, Raunak.
Hey.
Hi, okay. So our next speaker.
Okay, so you can see my slides, right?
Yes.
Perfect.
Okay. I'll announce here. So our next speaker is Raunak Basu from the Max Planck Institute for Brain Research, and he will be talking about the prospective representation of distal spatial goals in rodent orbital frontal cortex. Take it away.
So, thanks a lot, Anne-Marie, for the introduction, and thanks again for giving me the opportunity to present my research. So today I'm going to talk about the prospective and persistent representation of goals in orbitofrontal cortex. Now, let me start by introducing the navigation problem. So imagine this rat, right, located somewhere in the environment and needs to reach its goal. Now, to achieve this, the animal needs to calculate the optimal route from its current location to the goal location. But to do that, he needs precise estimates of both the current and the goal location before starting the journey. And in addition, the goal location needs to be maintained throughout the journey so that if there is an unexpected change in the environment, it can reevaluate the trajectory. Now, in the past 50 years, through the discovery of the famous place cell and the grid cells, along with a range of other spatially tuned neurons in the hippocampus and the surrounding cortical regions, we have a detailed understanding of how the brain represents the current location of the animal within an environment. So that answers the question, where am I? However, these cells fire only when the animal is at a given location, and hence, by definition, doesn't represent a location that is remote to the current location. And this brings us to one of the critical unanswered questions in the field. How is the goal location represented in the brain when the animal is situated elsewhere? So this problem is illustrated in the simple schematic. So during the goal-directed journey, so place cells will fire sequentially as the rat crosses the corresponding place fields. However, in parallel, somewhere in the brain, the goal location needs to be persistently represented throughout the journey. Hence, we thought that maybe goals are represented somewhere outside this hippocampal entorhinal system. And this led me to focus on the orbitofrontal cortex, a brain region that is critical for decision making and has been previously implicated in goal-directed tasks. And I recorded neural activity from the OFC using these custom-built tetrode-based recording devices. So to test goal representation, we designed a new navigation task where the goal locations varied throughout the session. The setup comprised a 2-meter long linear track with 10 equidistant reward wells that dispensed water rewards. And to obtain the reward, rats need to alternate between 2 wells at a time. So for example, here the rat drinks from well 1, then he has to go and lick well 8, then turn and go back and lick well 1 again. And this continues for 3 to 4 consecutive, continues unless he does 3 to 4 correct consecutive pairs of licks. So here's a video of the rat being trained in this task. So here the track is slightly modified, but the training protocol is the same. And you can see that they are quite comfortable doing this alternation. And yeah, and after 3 to 4 correct pairs, the rewarding wells change. And this change is signaled by switching on lights at the bottom of the track, along with water being delivered in new pairs of wells, which is well 4 and 9 in this case. And this continues for 10 to 15 blocks. Now, one important feature of the reward delivery is that animals need to lick a well for 1 to 2 seconds before the water is delivered. Hence, in a way, they need to commit to a well. And fortunately for us, rats actually learn this task relatively quickly, and they achieve stable performance within 2 weeks of training, as you can see from the average number of errors they make per block. Now let's look at some OFC neurons. So here I'm showing the activity of one neuron during single trials from 3 different blocks. So clearly this neuron likes to fire when the animal is approaching well number 2. However, if you just focus on this trial from well 8 to well 2, you'll see that as the animal crosses well 6, the firing rate is much higher compared to when the goal itself is well 6. So the firing is not very dependent on where the animal is currently, but rather it seems to be dependent on where the animal is heading. Hence, we turned to the activity of neurons aligned to the onset of licking. So here the trials are averaged based on the identity of the well that the animal is licking, and the wells are color-coded. And you can see that the neuron clearly distinguishes between different wells and fires highest when the lower numbered wells are approached. In fact, we found that the majority of OFC neurons have similar properties. So we asked that at the population level, can we decode the well that the animal is licking from? And to do that, we trained a LDA-based decoder and decoded the well identities in single trials. The plot here shows the average probability aligned to the lick onset over all trials from 18 sessions of data collected from 4 rats. And you can see that the decoding probability of the targeted well is quite above chance and peaks just before the animal starts licking. However, when I try to decode a well as the animal crosses it to go to a different target well, the decoding probability is much lower compared to what it was during licking. So this actually shows that the OFC neurons differentially encode well locations, but only when the well is approached as the goal. So next we asked that at what point during the journey is the goal-specific representation generated? Can we detect it as early as the beginning of the journey? And to answer that, let's go back to our previous neuron. So you have already seen the firing rate aligned to the lick onset. So now, in addition, I am plotting the activity aligned to the navigation onset, which is also the motion onset. So, and now you can clearly see that a very similar pattern of activity is visible at motion onset. And if I replot the activity of this neuron against normalized journey time, then the goal-specific firing rate is visible throughout the journey, like from the beginning to the end, right? And here's the example of another neuron, which also exhibits goal discriminating properties, but is much more dynamic. But you can see that the goal discrimination is occurring throughout the journey. So inspired by these neurons, we actually asked that, can we decode the goal well identity before the onset of navigation? And to do this, we devised a decoding scheme where the neural population activity across motion onset and lick onset, the subsequent lick onset, were merged together and fed into an LDA-based classifier. And the goal identity was used as the class labels. So using this classifier, we could predict goal well probability on held out trials. So this is an example of decoding performed on a single trial. So here the animal is navigating from well 8 to well 4. The y-axis shows the position along the track, with the well coordinates marked by ticks, and the x-axis shows the time relative to motion onset. Now the black line is the trajectory of the rat. So before t equals 0, the rat is at well 8, and after that he starts running towards well 4 and reaches there 1.5 seconds later. Now let's look at the decoded well as a function of time, which is shown in blue here. So it starts at well 8, and then just before motion onset, it drops to well 4 and more or less stays there throughout the rest of the trial. So here are examples of 2 more trials, and you can see that the decoded well abruptly switches to represent the goal location even before the motion onset. So here it's from 5 to 9, here it's from 5 to 2. So next we quantified this effect across all sessions, and what I'm showing you is a summary figure showing the mean goal decoding probability in blue as a function of time relative to motion onset. And the red curve shows the probability of the well that the animal is currently standing in. Now remember, this is the well that the animal just finished licking. So as you can see, the current well decoding goes below chance before motion onset, while the goal decoding is greater than chance almost a second prior to motion onset. And to further analyze the goal decoding along the entire trajectory, again, we replotted the decoding results against the normalized trajectory length and found that the goal decoding on average remains above chance level throughout the trajectory. So, and in addition, we also checked the probabilities of the well just after current well and just prior to the goal well, and observed that the goal well is represented much stronger than either of these two, thereby indicating that the OFC specifically represents the goal location, but not the intermediate wells. So till now, I told you that the goal representations form just prior to motion onset and is persistently maintained throughout the journey. But we return to the question of how this can happen. One way is through discrete point attractor kind of mechanism where each well is maintained through constant activity. However, given the dynamic nature of the neurons, we don't think that's very likely. The next possibility is through discrete but dynamic representation with similar dynamics between wells. Or the third option is when each well has their own dynamic patterns. So next, we actually look a closer look at the population dynamics to answer this question. So here, I'll show you a video of 2 seconds of neural activity from motion onset. And then there's a break, and again, activity from 300 milliseconds before to 200 milliseconds after the animal starts licking the goal. Now, this data is from one particular session where the population activity is projected onto 3, I'm showing you 3 PC dimensions here. And the activity is also trial averaged based on the goal well identity. So I would like to point out 2 things. First, the neural trajectories are segregated based on the goal well identity throughout the journey. And second, all trajectories have similar dynamics, but are only separated according to the well identity. And as expected, you can also see that the trajectories, the goal-specific trajectories sort of point towards their counterparts that are aligned to the lick onset. So once again, let's look at this in normalized time so that there is no break. And again, you can clearly see the 2 points that I mentioned, persistence of the goal and similarity in the dynamics. Now, these were trial average data. Let's see what happens in single trials. So I'll show you 3 time slices: motion onset, 1 second after that, and lick onset at the goal well. Now, using linear discriminant analysis, I projected the data across all trials on a linear subspace that best separates the neural activity according to the goal well identity. And then I have reduced it to 2 dimensions for visualization. Now here, each dot is a trial, and the average for a single well is shown with a big dot. Interestingly, the relative pattern of goal well encoding remained similar across time, although it went from a more compact representation at motion onset to a more expanded representation later on. Now, next, what I did was back projected these LDA projections onto real neural space and strung them together in time to produce neural trajectories that maximize goal separability at every time point. So now you can clearly see that even at single trial level, the encoding of the goal well is preserved throughout the neural trajectory, and that all the trajectories undergo similar kind of dynamics. So we asked that if we know the ensemble activity at motion onset, can we predict the trajectories for the next couple of seconds? And we used a linear dynamic model to answer this question. And you can see that the model trajectories look qualitatively similar. And to quantify how good the models are, we asked if we can decode the goal well identity from the model trajectories if we train the decoder on the activity aligned to lick onset. And we did it for all 18 sessions. And although not as good as the decoding the original trajectories, we can still see significant decoding of the goal well from these model trajectories. And so the OFC ensemble activity at the beginning of the journey contains enough information about the goal. So finally, we asked whether the neural activity is required in OFC is required for deciding the correct goal. And to address this, we expressed these excitatory options B reaches bilaterally in the OFC of 3 rats and implanted optic fibers just above the injection sites. And we could reliably induce firing of the neurons by switching on the laser. And we hypothesized that this would actually disrupt the OFC activity. So next we asked that if we perturb the OFC activity specifically during the onset of navigation, will the rats go to the wrong destination? And as a control, we also asked that what happens if we perturb it when the rat is licking? Will that affect the subsequent navigation? And we observed that indeed, when we perturb the activity specifically at motion onset, the animals went to the wrong destination 1/3 of the time on average. But on the control case, it was much lower. Okay, now to summarize, I've shown you that goal locations are represented in the OFC, and we can decode it on a single trial basis. And then I talked about how the goal-specific trajectories are embedded in the underlying neural dynamics of the OFC. And finally, I've shown you that optogenetic perturbation of OFC impairs the ability to target the correct goal, thereby establishing like a causal link between OFC and goal representation. So with that, I would like to thank the members of the Ito lab, and especially Hiroshi, who has been an awesome mentor and provided great support throughout the project, and my collaborators from the Tatjana Tchumatchenko's group, and my funding sources. And thank you all for listening, and I'll be happy to answer your question, and you can also contact me later in this email.
All right, thank you, Raunak. Very, very nice talk. We have lots of questions, probably not going to get to them all. So I'll just start with the top of the list here. Yu Liu asks, shouldn't the goal decoding equal to current location decoding at the end of the navigation? So I don't know when, i.e. when goal equals the current location.
Yeah, indeed, actually, and that's what happens. So that's why you see that the goal decoding probability goes much higher, right? So let me go back to the, yes. So here you can see that in the blue, this goes up. Actually, in the normalized position, that's much more visible. So the red, or what is the current, is once the rat has reached the goal, the red is the previous well, like where he started from. So yeah, so essentially what you is asking, it's true. So, but the nomenclature sort of changes once the rat reaches the goal.
Okay, and from Edmund Chung here, he asks, is there asymmetry in the responses depending on whether the animal approaches the goal from the left or from the right?
That's a very good question. Actually, I do have, so essentially, so yeah, so the OFC neurons do have some direction preference firing. However, what we saw that wells can be decoded irrespective of which direction the animal is approaching it from. So let me get to that slide. Yes. So if you focus on the figure on the left, this is showing the decoding ability of the goal well, or the well that the animal is approaching, where the training data set contains data where the animal is approaching from one side, and the testing is done on all the trials where the animal is approaching the well from the other side. And you can see that still the well can be decoded quite higher than chance.
Okay, and from Kenji Doya, how is this coding specific to the OFC? Is the activity modulated by reward amount or probability?
Yeah, that's also a very interesting question. We have not changed the reward probability or amount in this task, because we were just focusing on the spatial representation. And one way to think about it is, the representation is happening much before the animal is anywhere near the well. So the value is, the goal representation is occurring even without the animal having to actually lick the well, essentially.
Okay, I think in the interest of time, there's many great questions, but in the interest of time, we'll move on. And thank you very much, Raunak. Very nice talk. And I will call in our next speaker, Allison.
Okay. Bye.
Bye-bye.
All right, can you see me?
I can see you.
Awesome. All right, all ready?
All right. So our last speaker for this session is Allison Hamilos from Harvard Medical School. And she will talk about a slowly evolving dopaminergic activity that controls the timing of self-initiated movement.
All right, thank you so much. All right, today I'd like to invite us to think about an old mystery, the somewhat philosophical question of what makes us move. What goes off in the brain at a given instant that allows it to decide to spur the body into motion? Well, we know that when we move, thousands of neurons in higher motor centers begin firing together in concert, and this strong coordinated neural activity is relayed via spinal cord motor neurons to muscle fibers that power movement. But this isn't a satisfying explanation because, of course, it doesn't address why these neurons started firing together in the first place, which is really the crux of the issue. Now, one idea is that this activity could come from sensory areas. For example, if I throw this football to you, that strong activity charging through your visual pathways could be passed along to motor centers to trigger the coordinated activity needed to move. But not all movements result from overt sensory cues. For example, consider a cup of coffee sitting in front of you during this talk. Presumably, it's been sitting there long enough that it's no longer causing bursts of sensory activity in your cortex. Yet mysteriously, at some moment, your hand may reach out, pick it up, and take a drink. Now here it's not so simple to imagine where this motor activity came from. I will operationally refer to these two classes of movement as reactions and self-initiated movements, whereas self-initiated movement is distinguished as one in which the brain must rely on some internal process rather than an overt cue to generate it. It turns out it's not just intuition telling us there's something mysteriously different about these two types of movement. In fact, clues from human movement disorders also suggest these movements are generated differently. In particular, we all probably know a little about Parkinson's disease, a movement disorder in which patients feel slowed down and have difficulty initiating movement. But what might surprise you is that patients particularly struggle to self-initiate movements like picking up that cup, yet they can react with relative ease to sensory cues like a flying football. This so-called paradoxical kinesia is so striking that assistive technologies exploit it. For example, there are walkers that display visual cues on the ground to prompt patients when to take steps. Parkinson's results from the death of dopamine neurons in the substantia nigra, which suggests these neurons are normally vital to self-initiated movements in particular. So we asked, how might dopamine neurons be involved in self-initiating movement? Well, we know that the dopamine neurons lost in Parkinson's project densely to the dorsal striatum, where they exert a push-pull effect on the direct and indirect pathways. These pathways form parallel positive and negative feedback loops to the cortex. And one might imagine that these pathways could form a sort of cortical amplifier, with the direct pathway tending to enhance cortical activity and the indirect pathway tending to keep this activity in check. In this view, higher motor centers might be able to self-initiate coordinated activity when the gain of this amplifier is just right, or put differently, when the balance of the direct and indirect pathways is tipped in favor of the direct pathway. We hypothesize that dopaminergic activity could influence when movements initiate by tuning this balance, tending to push it toward the direct pathway, thereby pushing the system toward the moment of movement onset. To test this hypothesis, we recorded and manipulated dopaminergic activity in mice trained to make self-initiated movements. But to study self-initiated movement meaningfully, we had to create a scenario where the mouse had to rely on some internal process rather than an overt sensory cue to decide when to move. To satisfy this constraint, we trained mice to do a self-timed movement task, in which the mouse receives a cue and then has to wait for a seconds-long fixed delay before licking to trigger reward. Now, importantly, the animal is not being cued when to lick during this interval, but instead must estimate elapsed time to decide when to make her move. Now, because she has to rely on her own internal process of self-timing, these movements are implicitly in the family of what we have operationally defined as self-initiated. Well, mice, like humans, are pretty good at timing, but their timed behavior is highly variable. For instance, the mode of this animal's timing distribution is at 2.5 seconds, but sometimes note that he moves a second earlier, and other times he moves whole seconds later. It's important to note that this variability isn't a bug. In fact, it's vital to this task. It's exactly what we need to test our hypothesis, because with it we can ask, what are dopamine neurons doing differently on these trials when the animal decides to move early versus these other trials when he decides to move late? To answer this question, we took a fiber photometry approach to record dopamine neurons in substantia nigra, which is the population that's lost in Parkinson's. We specifically expressed the fluorescent calcium indicator GCAMP6F in these dopamine neurons using DAT-Cre mice and a Cre-dependent virus. This protein diffuses throughout the cell body and axon, allowing us to record dopaminergic population activity both at the cell bodies and at axon terminals, where these cells interact with the direct and indirect pathways. We also co-expressed TD tomato, whose fluorescence does not modulate in response to neural firing, but instead captures optical artifacts. Now, it's important to keep in mind that fiber photometry is not imaging of single neurons, but rather is an average fluorescence from all the neurons in our field of view. The mice completing the task are water deprived and head fixed, and they complete hundreds of trials of the self-timing task per session. On each trial, they receive a brief LED and tone cue to start their timing, after which the mouse has to decide when to lick in the absence of any further cues. The mouse's goal should be to make its first lick during the reward window, between 3.3 to 7 seconds after the cue, because if its first lick is too early or too late, it will not receive any juice rewards for the trial. Now, while the animals are behaving, we also monitor them carefully for ongoing body movements that could contaminate our photometry measurements, and I'll discuss these controls momentarily. But first, I want to just show you the most simple picture of what we found when recording dopamine neurons. This is an average of the GCAMP signal across all trials for one mouse where the response fell between 0.7 to 3.3 seconds. Now, the x-axis here is time, but there's two parts to the plot. On the left is a cue-triggered average where all the signals have been aligned to the cue and averaged together. And this is to show responses related to the cue. But I also want to show responses related to the movement. But remember, movements are happening over a very broad range of times relative to the cue. So I've split the x-axis here and plotted the average of all the trials aligned to the time of the first movement on the right. Now, the signals here are unfolding over the course of seconds. Let's start by focusing on the clear things we notice. We see there's a cue-related response, and we see there's a movement-related response. And neither of these features is too surprising. Dopamine neurons are known to fire transiently in response to salient cues and at the onset of movement. But what is surprising is the third thing we see, namely this apparent ramp between the time of the cue and the movement time. This signal starts several seconds before the movement occurs, and it appears as though dopaminergic activity is ramping up to the time of the movement. Now, the rising of this ramp made us think of a timer, so we asked whether this ramp was related to the mouse's natural variability in its timing. To test this idea, we pooled the GCAMP responses by the time of the movement relative to the cue. So here we're just looking at trials where the first lick occurred between 4 to 4.25 seconds, and we see the same rough patterns as before. Now let's add in another movement time. This purple trace shows all the trials where the first lick occurred between 1 to 1.25 seconds. Now we notice a few key differences. The baselines start from a similar level, but begin to diverge even before the cue occurs. The position at the cue onset is higher for the earlier trials, and the ramp does not dip down as low on the early trials as it does on the later trials. And finally, the early trials ramp is steeper than that of the later trials. Yet despite these differences, in both cases, the responses ramp to a similar level immediately before movement onset. It's important to remember that the differences between these two sets of pooled trials are the result of the animal's own natural variability in timing. We didn't change the task. Now, if we add in pooled trials that happen in between these times, we notice again that baselines start from a similar point, but diverge before the cue, after which follows a clear fanning of responses that already is predictive by eye of when movement will occur, starting more than 4 seconds before the movement onset, which is a long time for neurons. So just think about it. Just by looking at dopaminergic activity, we can learn something about what the mouse is going to do seconds in the future. We saw this pattern in every animal we tested. When we averaged together responses across all 12 of our mice, we found this pattern becomes abundantly clear. Now, here to more cleanly show the pre-movement responses in this plot, I've truncated the average traces at the time of the movement onset. By eye, we can see the dopaminergic signal is highly predictive of movement timing, starting even before the cue occurs, both via a baseline offset in activity, as well as a dynamic ramping signal during the timing interval itself. In contrast, the TD tomato control data does not show either of these patterns. We found similar dynamics in GCamp signals at dopaminergic axon terminals in the dorsal striatum, as well as in striatal dopamine release, suggesting these dopaminergic dynamic signals are passed downstream to the direct and indirect pathways, which are positioned to tune the timing of movement onset. Now, because these signals looked reminiscent of a timer, we asked whether a simple threshold model for GCamp could predict movement timing. And as our eyes tell us, this works very well. And threshold crossing time is highly predictive of movement timing, pretty much regardless of where you draw your threshold. You can draw it anywhere along this y-axis. Now, unfortunately, I don't have time to show these data right now, but GLM decoding models also showed that single trial GCamp signals were also highly predictive of movement timing, independent of task variables like previous trial outcome and ongoing motor confounds. And speaking of motor confounds, one key caveat to this picture is that dopamine neurons are active around the time of movement, and photometry is also prone to optical and mechanical artifacts during movements. So we needed to ensure that the timing-dependent ramping and baseline offsets we observed were not artifacts of ongoing body movements during the timing interval. To do so, we derived a GLM encoding model to isolate and quantify the contributions to the GCamp signal of factors like optical artifacts, behavioral events like the cue and the first lick, and nuisance body movements that might be happening during the timing interval, as assayed by neck EMG. This allowed us to distinguish contributions from these timing-independent factors from those arising from hidden cognitive processes related to timing of the movement, which we represented as a baseline offset, whose amplitude is proportional to movement time, as well as a temporal stretch feature, in which we represented percentages of elapsed time after the self-timed interval. Then, using a nested modeling approach, we first applied these nuisance movement and task variable predictors, which allowed the GCamp model to capture the cue and lick-related transients observed in the averages. However, nuisance movements did not produce the ramps, nor the baseline offsets observed in the GCamp signals. Conversely, these artifact predictors captured up to 80% of the variance in the TD tomato signal, showing that the model does indeed capture variance related to nuisance movements and artifacts. We then added the timing-dependent predictors to generate the full model, which robustly improved GCamp predictions. Importantly, the timing-dependent model was not guaranteed to find ramps when ramps were not present, as we can see in the TD tomato fit. The timing-dependent features also did not further improve the TD tomato model. We found similar encoding patterns in SNC axon terminals, as well as extracellular dopamine signals in the striatum. Now, given that dopaminergic dynamics were predictive of movement timing and were not explained by artifacts, we asked whether causally manipulating the activity of dopamine neurons on randomly interleaved trials would influence movement timing. To address this, we optogenetically activated or inhibited dopamine neurons on 30% of trials from the time of the cue up until the time of the first lick. Because dopaminergic activity had a higher baseline and ramped up more quickly preceding early movements, we hypothesized that activation might cause more early movements on stimulated trials compared to unstimulated trials. Conversely, we hypothesized that inhibition would cause later self-timed movements. To test this idea, we compared the distributions of movement times on stimulated versus unstimulated trials. We found that dopaminergic manipulation bidirectionally tuned the timing distribution, with activation causing early shifting, inhibition causing late shifting, and no opsin control animals showing no consistent effect of stimulation. Importantly, manipulating dopaminergic activity did not instantly trigger movement, nor did it prevent movement, suggesting dopaminergic activity influences movement timing by controlling the moment-to-moment probability of its onset. Taken together, these results argue that dopaminergic input to the dorsal striatum predicts and causally influences the timing of movement initiation. So what does this mean in the context of the basal ganglia? Well, the dynamic signals we observe do look reminiscent of a ramp to threshold process, wherein the rate of ramping depends on baseline dopaminergic activity. How a systems level threshold actually works in the brain is a little speculative, but we may imagine this could be the point at which the direct pathway activity critically exceeds indirect pathway activity, thereby permitting movement to initiate. We suspect that Parkinson's patients could live in a state much like the mouse when it moves relatively late on a given trial, but more extreme, in which the baseline dopaminergic drive is so low that even when patients make a big effort, the system can't ramp up very easily, requiring more time for the ramp-up process and sometimes failing altogether, resulting in delays in movement. To test this idea, more work is needed to understand how dopaminergic activity is relayed to and processed by downstream elements of the basal ganglia. We're excited to employ multimodal approaches, such as combining fiber photometry, optogenetics, and electrode recordings to simultaneously record and manipulate multiple circuit nodes to get to the bottom of what's really going on in the basal ganglia that makes us move. With that, I'd like to really sincerely thank and credit the team that's worked together on this project and all of you for being here for Cosyne 2021. And I'll just say that 15 minutes is definitely not enough time to really dig into the intricate and exciting questions at the intersection of timing and movement initiation. So I really hope you'll reach out to me and connect with me either by email or via Twitter or via GitHub here. That's posted as well. So now I just really want to answer some questions. So let's start our discussion.
All right, Allison, wow, really, really nice talk. Okay, you have a lot of questions. The top three actually seem to be quite related to each other.
Cool.
So I'm going to say this is kind of going to be a combined question from Tatjana Engel, Dieter Jager, and Yu Liu. And basically all of them want to know what you think is making these dopamine neurons ramp. Does it reflect single neurons, a population of neurons? Do the neurons, individual neurons themselves, have a different ramp rate? What is your opinion on this?
What a great question. I mean, it's total speculation at this point, because with our fiber photometry signal, of course, we're getting our whole population that we can view with our tapered fiber. So we don't know what individual neurons are doing, but I will say that there's been a lot of exciting work with these really deep implanted endoscopes, especially Alana Witten's lab, for example, that were able to examine dopaminergic activity from single neurons with calcium signals. So something, of course, we would love to do is actually look at single neuron activity. If we were to actually record with electrodes, that'd be a little bit tricky. SNC is a pretty thin strip. It's hard to target, but we could potentially do that with calcium signals. So what do I think? I don't know. It would be really, really interesting to find out. I think that you could imagine interesting mechanisms in which the whole population is ramping together, or individual neurons are turning on at discrete times, or if the population itself, the basal ganglia is this big feedback loop, right? Dopamine neurons are feeding into basal ganglia, and basal ganglia is feeding right back to dopamine neurons. So where does this loop start, right? Could dopamine neurons be revving up their own ramping as well through those feedback interactions? These are all really exciting questions that I would love to try to tease apart. So I'll just say that for now.
All right. Okay, from Grace Lindsay. She would like to know, would manipulating dopamine activity before the cue also have an effect, given that you see differences there?
What a great question. Okay, so I tried that, but I realized there was an important confound, at least in our task. We have a visual cue that is a light flash, and we worried that if we were stimulating neurons before the cue, especially in animals that might have gotten used to seeing some light before the cue, maybe they might interpret that stimulation as a cue-related signal. However, I do think that there's probably a way to do this that would be interesting to test out if you were able to use an opsin that doesn't have blue light feeding in for the stimulation. So, what do I think would happen? Yeah, I think that if you augment that baseline activity, I think there's good reason to think that you're going to speed up the pace of timekeeping on that next trial. That's sort of what it looks like in our data set, right? If you increase dopaminergic activity, you get faster ramping. So that would be my hypothesis. And we need to do a little bit further work to figure out a clean way to assess that.
Okay. So from Joseph Patton, he says, dopamine has been implicated in vigor as well as in timing per se. Do you interpret your data as reflecting dopamine's effects on vigor, timing, or are you agnostic?
Yeah, it's a good question. So, vigor has a lot of complex definitions, right? There's the one which is sort of, a goal-directed vigor, like I'm motivated, I'm ready to pursue a reward. And then there's this movement-related vigor, which is, tends to reflect movement kinematics per se, like acceleration, force of movements. I think that in our task, we have a couple things that we examined related to vigor. One question was, if our signals are actually vigor signals, you would expect that maybe on those early trials when the animals move relatively early, maybe the animals are moving more vigorously on those trials. That is to say, maybe the force or acceleration of those movements are actually somehow stronger. Well, I examined the EMG activity related to early versus late trials. I could not detect any differences between that EMG activity, which would suggest, at least as far as movement kinematics are concerned, we did not detect any specifically vigor-related signals. Now, to be fair, this is a tongue movement. Now, the tongue is capable of doing many complex movements, right? It can move quickly, it can move more slowly. So it is possible that you could have a more vigorous tongue movement, but it is fair to say maybe if you did a arm reach movement, you might be more likely to detect a vigor-related signal there. But I will just say that, we did look for those movement-related kinematic signals in the timing, in the differences of timing in our task. We could not detect those. So, I would say my impression at this point is that these are more timing-related signals than vigor-related signals. You could imagine an expanded definition of vigor in which you say that movement vigor includes force, acceleration, and also the timing of movement. But then, I think the difference between timing and vigor becomes somewhat semantic.
Okay. Well, I think that's all the time we have for this session. I would like to remind the speakers and our audience participants, there is the sessions session on the left-hand side of your hop-in screen, and you can go there and if the speakers are in their little rooms, you can ask additional questions there. And I strongly encourage you to use that option if you have really pressing questions, or find our speakers during Gather Town or any of the other sessions. So I want to thank the speakers for really, really excellent round of talks. And I want to remind everybody there is another session starting in a half an hour. We thank you for being here, and have a nice break. Thank you.